Okay, so this is the third and last video. We're talking about vaccine, but at the same time, we're going to be talking about a few other things like monoclonal antibodies, how it's useful. We're also going to be talking about allergies and how it works, and epidemiology. Okay, so that's another word that we probably have heard of quite a bit in the last little while. So let's get started. Now, vaccine is it has become somewhat controversial in the last number of years because of a lot of rumors saying about vaccine can cause autism or various other things. There's apps in there's absolutely no truth to all that. So what a vaccine is just a weakened form of the pathogen or just the antigens from the pathogen. Okay, so is usually dead or weakened bacteria. Now, with a lot of people who are immunocompromised nowadays, they a lot of times they will use the dead pathogen, or they will just use the antigen. Yeah, they will just use the antigens. They won't be using. They don't. There are not as many of the weakened ones as previously used. So either you inject it or ingest it. I remember one of the yeah, I remember before I went on a trip, I had to do a vaccine, which is by drinking it. So they will stimulate or stimulate a specific immune response, and so this process again, it's what you saw earlier. I'm not going to talk. You can read that part yourself. So yeah, so this is an example of artificially induce immunity. Now, in some cases, you might need a booster shot. So I think my hepatitis one is every 10 years, if I remember correctly. So there are some of them you need shots. That's why you need to keep a record of this thing. Now, because we're talking about vaccine, we have to talk about smallpox. And yay, this is one amazing things about why we do vaccination. Because smallpox was the first infectious disease that is eradicated. It's the first time that we managed to get rid of a disease, which is awesome and amazing. So it's highly contagious disease throughout the world. So measles. If you watch the video that talks about one infected person, how many people they would infect before they show symptoms or something like that. Right now, the coronavirus, it's I think one person will infect 2.2 or 2.5 or 2.3, somewhere along that line. It's more infectious than the regular cold or no regular flu. Measles was heck a lot more infectious. I think it was like 1 to 10. I think smallpox is even worse than that. But yeah, it's highly contagious disease. Here, yeah, kills, disfigured people who contact it. It's caused by this particular virus. Now, the vaccine is actually, you create the vaccine by, we're going to read the story here. So, Edward Jenner, he was actually a country doctor. So, he's actually a doctor that goes from place to place and treats people in farms, not in big cities. He was not that type of a doctor. He was kind of poor. Okay, so in this case, what he noticed that milkmaid, um, who has cowpox, which is a type of disease that comes from cows, would never get smallpox, which is much more dangerous. So in this case, cowpox. If you get cowpox, I think you just get a little sick, but usually you recover. Smallpox, you can die. Okay. So in this case, Jenner, what he did was he actually got parental permission. However, the child was below the age of consent. He got parental permission, but keep in mind, back in the day, like these were like back in the day, and you're a doctor, so people were like, "Oh, you're a doctor. We'll do whatever you say, sir." So in this case, it's iffy. But he infected a kid with cowpox. Kid recovered. Then next thing you know, he infected him with smallpox. Okay, and we found that the boy actually resists the disease. Okay, so Edward Jenner was the first person using human being as test subject and testing a vaccine. Okay, testing a vaccine. So the ethical complication. Okay, no previous lab research, no previous animal studies. You're talking about using a kid with a deadly disease. Please don't, none of you, please don't ever say random stuff like that as a possible IA topic. Outright, you say no. Okay. Back then, there wasn't really those rules, but the Nuremberg trials at the end of World War II, because there were a lot of horrible things that have happened in the name of experiments. So in this case, scientific experiments. So in this case, from Nuremberg trial, they establish a code of conduct and ethics. So you is supposed to protect human research subjects. So right now, if you think about it, this kind of thing would have never been allowed nowadays. Back then, you're talking about 1700s. Canada even wasn't even a country. I don't even know what's the history of US. So yeah, that's a very different world. Anyways, back then. So the cowpox, the vaccine is from a virus called cowpox. Now, what they did was there was a campaign by World Health Organization back in the 50s. So what they do is they identify people, they isolate them, and then they will kind of trace their contact. So who has this person been in direct contact with, and all those people were vaccinated. Okay. What they found is that the infectivity was only 3 to 4 weeks. So basically, people who have the smallpox, they are only infectious, they'll pass the disease for 3 to 4 weeks. And then afterwards, they live or they die. No carriers, so recovered patients did not retain any virus in the body and no animal vectors. So this is actually one of the condition about smallpox, which allows it to be eradicated. So it's not like, oh, this we healed all the human, there's no more cases of smallpox with human, but it's somehow, let's just say, in bats or in monkeys, and the next thing you know, it comes back a few years. Thankfully, no, smallpox only infect human. There's no animal vector, so it was okay, and we don't have this whole carrier thing, which is like the issue that we're dealing with COVID-19 now. Also, another condition for smallpox, which allows this to happen, is that it is a stable virus. It doesn't mutate. So in this case, it doesn't change the surface antigen. So the memory cell will always recognize it. Okay, and again, it relies on international effort and cooperation. So they would lead to a successful eradication. If it wasn't for these conditions and this practice, smallpox wouldn't have been eradicated back then. It was been eradicated since 1977. Um, Basically, the only places where there's smallpox right now are in high security level government labs. Okay, so there are some in Europe, in Russia. There are a few in U.S. CDC and the U.S. military. I think the Canadian laboratory in Winnipeg have some maybe, but yeah, that's it. And it's always under lock and key because it is so infectious. And since we eradicate it, we don't vaccine people for smallpox anymore. So in this case, everyone on Earth is, aside from some lab scientists, people who work in the lab, pretty much everyone are susceptible for smallpox. But because we don't vaccinate them anymore. Okay, pause this for now. Any questions? Been talking for a long time. Okay. Okay, monoclonal antibody. Basically, on a monoclonal antibody, what you use is you actually can use it for diagnosis or therapeutic. So you can actually use it to treat disease. You can also use it for detect things. Okay, and we're going to talk about what they are. So now, when you talk about monoclonal antibody, we're talking about only one type of antibody. Okay, whereas polyclonal antibody, basically what your B cells will be doing, respond to multiple antigen. This is responding to only one type of antigen. Okay, so you have more than one B cell involved. This is just one type of B cell. So in this case, you're essentially making a hybrid of a plasma cell with a cancer cell. Okay, so fusion of a tumor cell with a plasma cell. So this is what it's called. So you're fusing it. The tumor cell will allow, if you remember your cell biology, the tumor cell will allow the B cell or this this new cell to keep on multiplying and keep on producing itself. Also, at the same time, as long as it's given a nutrient, it's not going to die. Okay. Um, and the B cell will keep on making antibodies. Now, these antibodies purified and produced and can be used. Now, what's so amazing about it? Pregnancy kits. Okay. Now, before this home pregnancy kit was created or invented, or ladies back in the days, they only really know they're truly pregnant by going to the doctor, where the doctor will do a pregnancy test, which is pretty much the same kind of deal. Okay, they will draw blood and they'll test for the antibody. They'll test for this particular protein. Okay, however, home pregnancy kit changed all that. Okay, nowadays these pregnancy tests are highly reliable. I don't remember whether it's 95 or 99%. It's cheap, it's easy. And in the past, I actually get a showed do a demo with that. So how does that use? So pregnant women produce this type of protein, which is present in the urine and indicate pregnancy. Pregnancy kit is actually a process called ELISA, enzyme-linked immunoabsorbent assay, to identify a substance via a color change. So what they do is you get a urine sample from the ladies. So the idea of peeing on a stick. Usually it's the first pee in the morning because that's the HCG concentration in the urine will be the highest in that first pee in the morning. Once the pee sample got in onto this stick here, this will absorb. This is an absorbent material. So in this case, the urine as well as any of the HCG will move up along this along the pregnancy test. So you have two lines. Okay, you have two lines. The first line here is to show. The first line here is actually the test region. So it's actually you have the immobilized antibody. You have immobilized anti-HCG antibodies. They will bind onto the HCG itself, so it will be trapping. Oh no, sorry about that. So the first part here, you have any HCG will bind to the first HCG antibody. So it's actually there's antibody to the HCG here. This here this site, you actually have a different antibody will bind to the separate part of the HCG protein itself. And there's an enzyme which will cause a color change on the coating. So that's a first line. So first line is a positive test. Now, the second line here is these antibodies are not specific to the HCG, but to actually the antibody themselves. So this line here is to just really show that it will again cause a color change. So it's just a control. So if you're pregnant, you'll see two lines. If you're not pregnant and the kit is working properly, then you will not see a line here, but see a line here. This line shows that this test is working properly. But if there's something wrong, like this line is not showing up, something is off. Okay, try again. Pause. Any questions? Sometimes occasionally they do ask this question. Okay. So allergic reactions. So the white blood cells, the white blood cells do release histamine. Now, histamine is another chemical signal. Okay, another chemical signal. Usually, it's when you have inflammation. Okay, so histamine will be released from white blood cells. There's a type of white blood cells called mast cells. It will cause increased blood flow, increased permeability of the capillaries, increased accumulation of white blood cell and plasma. So the site become swollen, warm, red, and painful. Now, if you read this, that's inflammation. A part of your body that's swollen and painful and red, that's because of a localized response to a damage. Okay. So here's a diagram here showing all that. If you have a pinprick or something like that, bacteria coming in, chemical signals, this is happening. Now, an allergen is essentially a substance from the environment. It's would trigger immune response, but it's not harmful. So the immune response tends to be localized to the area of exposure. That's why you will cough and sneeze, and basically the area around here. Very severe ones will have anaphylactic shock, can be fatal if not treated. That's why in that case, if you some of you might need an EpiPen. So that's what you do to slow down your immune system. It's straight up, not steroid, straight up epinephrine, so which is adrenaline. Allergic reaction require pre-sensitized immune state. So you need to have prior exposure to the allergen. So in this case, if you never got stung by bees before, you would not be allergic to bee stings. The tricky part is, do you remember it? If it happened to you as a baby, you then might not remember that. Okay. So again, this is very similar. If you read this again, it's very similar to your first immune response or first specific response of your antibody. But right now, instead of thinking about antibody, you're talking about histamines being generated. Okay, last but not least, epidemiology. So it's a study of incidence, distribution, and possible control of disease. So right now with COVID-19, all the epidemiologists are working full-time and whatnot. Basically, they're studying how different spread of the disease is and where it will go. So vaccination program led to reduction of incident of infectious disease. That's why make sure you get vaccinated. One really glad thing from this COVID-19 thing is I'm so thankful I have not heard anyone who are the anti-vax people saying anything. Thank goodness. Um, Different country implement different vaccination practice depending on what their local regional pathology threats are. That is true. Like when I lived in different countries, my children who were born in different places, they all have different vaccination program card. Granted, we got them to follow the Canadian one, but when we're living overseas, we follow the local one. Now, the record of these disease and immune system can be are updated and can be accessed through the WHO data and statistics, and that's the website. Some of you might want to do that as a database IA. If you're interested, we'll talk about it. Okay. You can use to compare the incident of disease over time or compare the incident of a disease in different region. So in this case, you're talking about before, after a vaccination program, or both with or without a vaccination program. And in some ways you can talk about how COVID-19 right now and how spread out they are in different countries. Now down here is a case study on measles. Now, why are we talking about measles? Because for a long time, there's a misconception to the measles vaccine, and IB is working quite work. IB take that into account. Measles is highly contagious, and sadly, easily preventable. But because of the anti-vaccination group, people have not done it. So what I'm gonna ask you to do as homework, is I would ask you to try to go and do this case study on the on the on the measles and about their vaccination program. Um, that is for homework, not due next lesson. So not you don't need to get it done by Friday lesson, Friday or Monday's lesson, but you can do it after the test. Okay. Oh, the lesson after. The lesson after. Okay. So that's been a lot. If you have any questions, make sure you type it into the puzzle part. I'll look through it and then I'll respond to it. Okay. Have a good day.